Again, good morning, everyone. As Heiner was saying, so we are for a couple of years now pursuing in my working group in Bonn the idea of using electrical methods, electrical measurement methods to investigate roots, with particular view to root structure and root function. And I was very happy when it turned out that we could be integrated in this FINROP consortium with our research. And today I would like to try to give you an overview of the concepts and principles actually behind that. So we've seen already some results, different occasions, and hopefully you'll get some more insight after my talk today. I would also like to acknowledge my co-workers here, in particular in this FINROP project. This is Maximilian Weigand, a postdoctoral researcher in my group, and Valentin Michels is the PhD student here in our sub-project in CP3. And then from Forschungszentrum Jülich, Egon Zimmermann and Sander Heusmann. In particular, Egon Zimmermann is a very, very important contributor with regard to the measurement devices that, as you will see, we will need for our research. Yeah, I start with the general motivation, which is quite obvious. There is great interest in the availability of non-invasive tools which have the capability to image root structure and function at the field scale. And so that has been driving us for a couple of years, as I was saying. And you see this image that was shown also by Andreas recently in the advisory board meeting, where this is measured at Klein, or an imaging result obtained at Klein-Altendorf, where the idea is that with electrical properties that we image, we are able to, on the one hand, delineate root zones, and on the other hand, by really quantitatively analyzing the values actually in these pixels or voxels to also infer information on root structure and function. And for this, we use the methods sEIT, which you have seen before several times as well. And this stands for Spectral Electrical Impedance Tomography. And in a way, this already captures what I will talk about today. So it's a combination, the methodology is a combination of spectroscopy on the one hand and tomography, so with an imaging modality on the other hand, and that based on electrical properties and more explicitly electrical conduction and polarization properties of the soil-root continuum. And of course, it's important to look at soil and root properties individually to also understand these signatures. And that's what I'm trying to do today. The following way, I will first briefly address electrical properties of soils and roots, because I think that's also important with regard to our contributions, our work here in the cluster. Then I will recall the basic principles of the imaging algorithm, or the impedance tomography method, and then show you a number of results that we obtained in recent years in lab and field studies, and of course ending up then with what we've just seen in the slide before. Okay, to start with, electrical imaging has been used in geophysics for almost decades, let's say 20, 30 years, with the availability of computational resources and the measurement principle. So measuring electrical resistances or impedances goes back to already the beginning of the last century. And the idea is to inject the current into the ground. So this would be then this would distribute within the subsurface depending on the electrical property distribution in the subsurface. So the current inflow is indicated here by the green solid lines, and associated with that we have an electric potential field, that's the dashed lines, and that can be measured by additional pairs of electrodes. And importantly, the pattern of electrical potential is directly reflecting the conductivity structure, also polarization structure to some degree in the subsurface. So this way the information on the subsurface electrical properties is collected. And by doing that now in a way surrounding a target region by as many electrodes as possible, and then systematically varying this 4-electrode measurement setup, you can collect basically a set of resistance or impedance values, that is measured voltage over current, and apply tomographic inversion algorithms to turn all these individual measurements into a single image, 2D, 3D of the electrical properties. And the whole theory and modeling is implemented in a continuous macroscopic manner. So once it comes to the interpretation or analysis of these results, of these pixel values, there has to be understanding of how the effective properties that are imaged at the macro scale, how they actually generate from the microstructure and processes in the porous medium, or also in the root porous medium, potentially including roots in our case. Okay, and so this was for the geophysics. So that's been investigated and developed for decades, as I was saying. And on the other hand, it's also been known for a long time that that roots come with certain electrical characteristics. So here, for instance, showing schemes from an earlier work here, where you see the electric current patterns or electric field patterns associated with root growth at the root tip. And the electric field that is generated basically, or that you can see here, reflects also electrical charge distributions. That's fundamentals of electrostatic theory. And as we will see, the idea is seems to be straightforward, to also using these electrical characteristics, properties within roots, around roots, in an imaging framework by actively measuring those. Yeah, and this maybe as some introductory words on where we started. And now we look into the electrical conduction properties, first of soils or soils and rocks as porous media. And as many of you will know, is that the bulk electrical conductivity of a soil for reasonable water, or for sufficiently high pore water electrical conductivity, so let's say salinity, basically the bulk conductivity scales directly linearly with the salinity. And this linear regime is sometimes also, or typically referred to as the Archie region, where then also petrophysical parameters like saturation and porosity and some empirical parameters here determine this proportionality between the two. If salinity in the volumetric pore water is decreased significantly, then actually this goes into an asymptotic regime where it's dominated by another conduction mechanism which takes place directly at the mineral interfaces, so the mineral pore water interfaces, and which is effectively independent of the salinity in the volumetric pore water. And this is normally an effect of pronounced in clay materials, where we have certain minerals that come with a high electrical surface charge. And assuming that these two, let's say the volumetric path through the pore water and then this interfacial path along this interface, act electrically in parallel, we can just sum up the conductivities of these individual mechanisms and come up with an bulk conductivity. So that's basic electrical models describing the quite well-known phenomena. And if we zoom in now into the porous medium, and all this I should say also assumes, which is typically the case for soils, that we can neglect electronically conducting minerals. So this again, otherwise there would be an additional mechanism to consider. If we now zoom in into the pore space, into this interface, so where of course the current is manifested, is the charges are transported by an external electric field, then we see cations and anions build up accumulations. So ion concentration gradients in either the vicinity of pore constrictions or maybe around mineral components within considerably large surface charge. And yeah, so this is actually the result of charge transport in an external electric field. And that's what we do. We apply in this sEIT method, we apply an electric field to the porous medium. And once we turn that off, it's quite obvious that the system that is where we have these concentration buildups, that relaxes again into some equilibrium situation, which basically is referred to the well-known electrical double layer scenario. And associated with that, we also have so-called polarization currents. So this is basically a capacitive effect, which means polarization, and this, as soon as we turn the electric field off, this fades away. And so this effect, this mechanism in the soil or in the rock can be measured and described, of course, in particular in the frequency domain by not only a ratio between the current amplitude and the voltage amplitude, which basically defines the resistance, but also gives rise, the polarization here, to a phase shift, so to a delay of the voltage response to an injected current. And this phase shift, let's say for an harmonic frequency here, can be measured and captures the polarization at a particular frequency. And mathematically, this can be conveniently described by extending the scalar property conductivity to complex valued property, complex valued conductivity, or just shortly complex conductivity, comprised of magnitude and phase. And actually also with frequency dependence in it. So the single parameter that most of you will know, like let's say soil electrical conductivity, actually becomes a complex valued spectrum if all these if the full nature of these properties is considered. And typically this can be then also separated into surface effects, so referring to the attributed to the surface conductivity that I was mentioning, and to the electrolytic conductivity according to the Archie regime. So, so basically that is the target property also for the imaging. It's a complex value comprised of magnitude and phase, and it has a frequency dependence. So to measure it, it requires varying the measurement frequency. And in some way, it contains information on the microstructure of the soil, of the porous medium, so the texture, the mineralogy, so with the surface characteristics at the mineral grains, and also the pore water chemistry and saturation. So it's a wealth of information that can be inferred from these spectra. And and over the last 10 to 15 years, there's been many studies on much work in that direction in the field of near surface geophysics. And one important or remarkable phenomenon, also principle in terms of conceptual understanding, is that polarization mechanisms at different length scales in the microstructure actually show up in the spectrum. Here is now resistivity plotted, the inverse of conductivity. Here is the phase shift spectrum. So these show up particular length scales at particular frequencies, directly being related to, of course, certain relaxation times, so inverse of frequency. And if we have several of these mechanisms, these can add up, superimposed to an effective spectrum, which can be also mathematically described by, at least phenomenologically, by just adding up appropriate dispersion terms. So very common is they are just superimposing, adding up in responses with individual relaxation times corresponding to individual length scales in terms of these Debye or Cole model forms. This is just kind of the functional form that you see here. Again, this is a complex transfer function, I being the imaginary unit here. So complex transfer function between injected current and measured voltage, basically. And the important parameters in these phenomenological descriptions are chargeability as a measure of strength of polarization. so shifting the red curve vertically here, and relaxation time here, which determines the frequency where these peaks show up. So that basically controls the response in that direction. So that's a very easy way of describing phenomenologically what we observe. And now looking at first measurements that have been done on plants, including roots, we see that actually here is the polarization properties measured with a simple 2-electrode setup where the overall current is basically driven through the soil, through the whole root system, and then through the stem and back into the device. So what is measured here is sort of an averaged or effective signal with contributions from soil, root, and particular different zones also, or different elements of the roots. Nevertheless, and this is in a frequency range, you see a 1 kilohertz, 100 hertz to 10 kilohertz. There is this relatively sharp peak that is reflecting polarization properties, and it has been observed that this polarization amplitude increases with actually the growing of the plant. So with actually the increase of root mass also that is measured or sensed here. And based on this, the hypothesis came up that okay, and also now considering the conceptual understanding that has been developed in the geophysical world, that basically, again, relaxation time, so the position of these peaks, capture root size information, so related to root size distribution, maybe even root architectural parameters, while the magnitude actually in that direction is related to root volume, root surface, root mass. And in particular, as we will also see then in experiments, which I will show, also the activity of the root. So it's not just the presence of the root, but also the state of the root that matters here. And yeah, okay, so that's the basic idea, understanding actually what is included here in the different ways, and then putting all this in an imaging context and doing that at the field scale. That's basically the idea, and we're heading for that now for a couple of years and already made a few steps forward. So this shows measurements 10 years ago already, where you see now measurements, these impedance measurements in terms of a phase spectrum on an individual root system in these, like you see here, for different solutions where the root was placed in. And what is interesting, or was interesting at this point, that there seem to be like two different polarization mechanisms that you see here is reflected by the shape of this spectrum. And of course, we can now quantify such responses in terms of models that I mentioned before. And here considering that actually time scales where these responses occur, and also considering basic physical relationships given the diffusion character of movement of ions that of course play a role here in electrolytes, either in soil or also within roots, it is suggested that this higher frequency response basically is caused by polarization properties rather at the cell root scale, so cell scale of the root, while this lower frequency, so slower relaxation or polarization process, is related more to the macro scale of a root, or maybe individual root, the whole entire root scale. And that is kind of still open questions today that we are also pursuing. One approach, obviously, would be the brute force modeling of these responses for what is known. And I mean, much is known about root structure and also electrical properties of roots. So one could try to basically forward model these responses. And that's been done in the past years, quite recently also for rocks and soils. So where then really process-based, the dynamics of ions, cations, and anions around, for instance, spherical mineral grains is modeled using then corresponding physical equations. So the dynamics is described here by the Poisson-Nernst-Planck equations, or system of equations for cations and anions. Basically, that reflects charge conservation with here diffusive ion fluxes and electromigrative ion fluxes, so transport of charges in ions in the electric field, both coupled by here the Poisson equation, basically. And so this is just to give you an idea or an impression. So basically, one could translate that also in the world of a root, taking the structure and so on, and implementing this in finite elements and so on, and then go straight forward and look which elements actually are responsible for which characteristics in the responses. And this analogy between the mineral surface characteristics and the root cell, or let's say root membrane characteristics, actually led us quite recently also to the hypothesis, which is shown here on the right, where you basically see the profile of the electric potential across the plasma membrane, where we have this given a potential gradients and a potential difference between the root cell basically and the exterior. We have the buildup of electrical double layers, which are a key element in the buildup of polarization once this sees or experiences an electric field. And with the transfer of ions, so let's say flux of ions across this membrane into the root, basically, also the potential drop changes and gives rise actually to change also in the polarizability. In this case, actually, it decreases because kind of the voltage gradient here, this drop, which is now important for the polarization on that side, is smaller than on that side. So this is just to give you an idea where basically we would have to go and which kind of models and theories to adapt if we want to at least process-based physically or physico-chemically understand these responses. Not talking of course about the upscaling that is needed at this point to the root system scale. Okay, and from this is important to note, we also expect that our electrical polarization response also at the whole entire root system scale bears some sensitivity to nutrient uptake, actually. We don't know exactly where, at which scales all this happens, but there should be a general sensitivity. So which is important for us because this is at the basis of what we would refer to as physiological sensitivity of this electrical measurement. Okay, now you may ask yourself, having seen examples for root responses and soil responses, these somehow mix up in the real field situation, so how can we discriminate those? And for this, I would like to show you this simple study where we have on top these conductivity spectra, real component and imaginary component for a soil sample taken from Selhausen, actually for different water saturations here. So we see the increase of water saturation of the real component, which we know. We also see, of course, we see an effect also in the imaginary spectrum. This, on the other hand, is the response of a root. In this case, I think it was winter wheat, if I'm not mistaken, which if you now compare the values here, this is orders of magnitude larger in terms of imaginary conductivity, so polarization strength, than actually what we have here for the soil. So that is, of course, one important aspect. And now if we actually mix those and so upscale this to a two-component response, if you want, we can adapt methods like power law mixing, which is kind of an approach used in different areas where we basically take the individual responses, and here you have to see again, this is complex valued spectra behind, We have soil, we have root, and we have a volumetric fraction of root, which is of course relatively small in our case, and a volumetric fraction of soil. And then we add things up in using an exponent here, alpha, which is of empirical nature and basically scales within or ranges between -1 and 1. And from this actually goes from these two components being in parallel to these two components being in series. So if we do that to get an effective response, then we see basically that looking at the different water saturations, that the effective response, that the effect of water saturation in the effective response is mainly visible in the low frequency real component, in the low frequency electrical conductivity, while the effect of changing the volume of root in this effective properties is most has the strongest effect in the high frequency polarization component, in the high frequency imaginary component of conductivity. And so this is what we think is the key. That's why I'm showing this and probably spending too much time on this, but I think it's important, and it's sort of the key also for then accessing roots, extracting root informations from field measurements. So we have to have spectral measurements. We have to actually use the spectral behavior of these signatures. Okay, maybe last word in this context also on this parameter alpha, because we did some interesting work also on that. There is reasons to assume that this actually depends on this empirical parameter and varies systematically for root systems with root system architecture and also direction of current flow. So whether, for instance, the current runs or flows vertically or horizontally through this root system. And one way to investigate that numerically is by, again, brute force simulations, by setting up, which actually Satyan Rao, a PhD student of Mathieu Javot in Louvain-la-Neuve, where I was also involved, which he set up using C-root box, so different root architectures and so on, and then applied different spectra to root and soil, and then modeled actually the effective, the macroscopic responses in both directions, so horizontally and vertically, and then ended up with spectra like this. And of course, now the idea is to understand the meaning of alpha in terms of given root architecture, and it's quite interesting. So first of all, actually this simple macroscopic mixing model could be fitted to the explicit forward modeling results here. And it turned out that, as I was saying, we need, of course, for the conductivities, then complex conductivities in the different directions, different values of alpha, because that's also the way they act in the model. And which I will mention probably in the outlook, basically requires that the whole imaging is set up for anisotropic parameters. So not only complex valued and full frequency dependent, but on top even yeah, in an anisotropic way. Okay, so now coming to the principles of the imaging, and that's, I think I can go through that a bit faster, is I already mentioned that the forward problem is driven by a Poisson equation. So we have electrical conductivity, we have electric potentials, we measure these ratios of potential differences over current, which if it includes phase shift, that represents an impedance, and these are using tomographic inversion schemes inverted into a complex resistivity or conductivity spectrum at pixel or voxel scale. And behind all that, that's why I wanted to mention that, because that's what you don't see normally, but behind that is really the solution of the non-trivial solution of a non-linear, non-unique and ill-conditioned inverse problem, which we normally tackle in a standard way by minimizing an objective function where we have like a data misfit, data minus model prediction, forward model prediction, weighted if we have data uncertainty information available, and then with a regularization term added, which normally represents the roughness of the spatial distribution, so the spatial roughness. So once we do this, when we minimize that, that makes the inverse problem unique in a way. So it's required, this sort of regularization strategy, and typically this leads then to relatively smooth models. And by solving this objective optimization problem, this leads to the well-known sort of normal equations where yeah, you see something like this here, which still is, these individual entries are all complex. And also basically we have these equations for each considered frequency. Okay, and then we have this, we can turn these measurements into images, which typically, and this is shown in this simulation, based on also simple models, synthetic models of root architecture, which come with some spatial resolution limit, which is inherent to the physics, to the basically the sensitivity associated with these electrical measurements. On top of that, of course, also the smoothing effects due to the aforementioned regularization. So I'm saying that because it's important to know that we cannot expect that we'll actually spatially resolve individual kind of root segments of a root system with this method. That will be impossible. But the way is actually to, of course, know the spatial resolution and now interpret the spectrum at this scale in terms of root architectural characteristics, as I was saying. So the information comes from the spectrum, not from the spatial resolution. So if you want, from the spectral resolution, not from the spatial resolution. That's inherently limited. Okay, so if we do that, then basically that's again illustrated here, what I just said. And it's important to note, as I was saying in the beginning, that we need measurement devices to collect these data, and it requires multi-frequency data acquisition, actually as broadband as possible. So typically we use frequencies from 10 to the minus 2 hertz to tens of kilohertz. 10 to the minus 2 hertz has restrictions again in terms of measurement data acquisition times. If you think, for instance, of monitoring, because that would mean like 100 seconds basically for one period, and you need several of those, so that sums up to very huge data acquisition times. And on the high frequency end, things are sort of limited because of particular electromagnetic induction effects, which are physically there, and which, if we want to do it properly, would have to be even included in the forward model. And it's perhaps possible at some day, but not today. And the other thing is actually the phase accuracy that is needed, and I didn't emphasize that before. Actually, the polarization is very, very small. So these phase angles are very, very small. That is much less typically than for soils than 1 degree. So it's normally measured in, or given the values in milliradians. So it's very small and needs corresponding accuracy. And fortunately, we have available also here in Phenorob systems, or one system for now our field measurements that we have developed together with the colleagues in Jülich in former times, and which is actually designed to serve actually the purposes and requirements for these type of applications. So it's actually not possible to buy a system like this. So we are lucky to have that available, and otherwise the research would not be possible, at least for the experimental work. Now coming to the applications of the method at the different scales. So also I would like to let you know that we, as you could already see, that we measure individual root segments. So we basically try to improve our understanding of what is really controlling the properties of the individual root. We do Rhizotron experiments, or we did, we still do, And that's also where we started a couple of years ago with plants in nutrient solution, also with soil. And of course, we have field measurements like now in phenorock. Yeah, so to give you one example of the measurements in the lab and the Rhizotrons without soil, actually we tried it first with soil, so it was much too ambitious as it turned out, and we had to go back because we were not at that time knowing the right analysis procedures and so on to actually to discriminate the soil and root responses. So this is only with the root system, in this case an oilseed plant in some nutrient solution. In this case, it was just tap water, actually. And you see the images that you get at 1 Hertz and 70 Hertz in terms of conduction, real conductivity component, and in terms of polarization, imaginary conductivity component. And this just shows that with increasing frequencies, it seems to be a better possible to delineate the root system, in particular in the polarization signature. And from the type of spectra that I've shown before, I mean, this is what we can expect. So we have this higher frequency contribution compared to the typical responses of soil. If we now, so this is the imaging information, if you want, that we get from these measurements. Now, if we look at selected pixels, really at the full spectrum, and in this case, for the same experiment now over time, so in a monitoring mode, we see here 2 pixels which are taken from the root zone, so where roots were present. And here's 1 pixel for comparison from the lower zone without roots, so just tap water. And then gray basically indicating where we run into measurement accuracy issues. Yeah? So we see that basically here we think it's quite reliably measured. And here we see clearly the root response, which now from blue to red fades away over time. And this actually goes along with the continuous degradation of the plant. I didn't put pictures here of the plant, but you could, of course, see it also given the increasing nutrient stress or deficiency, since there was no adding of nutrients at any time in this experiment. Yeah? So this kind of is also further evidence for this physiological sensitivity of these responses that we tried to image. So polarization fades away. And another experiment, the results of which I would like to show, you might have seen that on previous slides and some of the general talks, is the now without imaging, because we wanted to have high temporal resolution, is just the polarization property that we measured, in terms of some sort of chargeability, but that doesn't matter in this case, during ongoing artificial day-night cycles. So this is, I'm sorry that you cannot see the axis. So this is actually time, and a gray bar, or also the light gray, dark gray bars, each indicating 12 hours. So this is basically artificial day and artificial night, and then in cycles, with some growing lamp and so on in our lab. And you see the cyclic behavior also of the response, which is indicative of the effect of the physiological processes in the polarization properties of the root. And interestingly, also we had this, just for the sake of curiosity, we came up with a what is this, 24 hours or even longer night, if you want. And you see, that was interesting. There seems to be some memory effect that we see here in the root. So it first kind of starts going back again in this direction as it's used to, but then something seems to be different, and then actually runs again into this behavior which we observed, so an increase of polarization which we observed during night. And of course, that is quite promising with regard to now interpreting also these type of monitoring data in an imaging context in field experiments, where we have then, of course, independent measurements and information to, for the interpretation and of these data. This brings me to the field, and I think that's now the last 2-3 slides. I hope I'm doing okay in terms of time. So you've seen this. This is the Selhausen Rhizotron facility with an linear array of electrodes and some electronics with the electrodes here across three basically plots here, which have been treated in a different way. Here you see the measurement system in the basement here of the Rhizotron facility. And now this is the result that Andrea has also shown at the advisory board meeting, where from the spectral responses that you've also seen now before, we figured out that basically the ratio of the imaginary conductivity at high frequency to or over the imaginary conductivity at lower frequencies, and where we pick those is actually not does not matter that much. That actually seems to capture nicely the change of the spectral shape of these responses due to the presence of roots. So that's why we just should choose those as a first preliminary indicator for presence of roots. Because at this point, I mean, we would need really the models to actually do better than this. And it turns out that if I now also start this animation here, maybe stop it. Here you see the vertical section through these three plots at the Rhizotron facility. This was in 2018, where maize was grown, and at these different plots here, actually in double density in the middle. And then we had like a shelter plot and an irrigated plot, so different water content states, if you want, also associated with that. And this indicator here, this value here is now, based on our current understanding, it should indicate presence of root, and in particular with increasing values, also showing increasing root surface area. Let's say that would be the value. And if the next slide, that's compared to the root length densities that are derived from optical measurements. First of all, I want to say that, also this would make sense since we have a bit darker, higher values here where we also have this double density mice. Right? So we would expect more root mass actually in this for this middle plot. And then we also, if I continue with the animation, then you see here that's the colored lines are actually the evolution of this value in different depths. And you see that actually for the shallow depths, it increases first, right? And then, okay, at some point again, decreases, while then with increasing depth, that's delayed, right? So that obviously would reflect also the growth of root into the subsurface as we expected. And if I now compare or show you here the comparison of individual vertical profiles for the three plots here for different times, so with ongoing root growth from blue to red, then we see, for instance, that we have the maximum here, this peak in these red curves. So at later times, this peak is shallower than here, for instance, for the left plot, which actually could be explained since this was irrigated. So maybe there was more water available and the root was not forced to actually grow into the depth like in these plots here, in particular the left plot. And if we compare now this with the results that we got from the optical images in the rhizotubes, then we basically see nice correspondences in the qualitative behavior of these data, both over depth and over time. So this is, as we think, quite promising. And that's also the reason why I think that Valentin, now our PhD student pursuing now these, the method in particular at the field scale, is probably excited also about this result that was also shown then the advisory board meeting. And here in my first slide, where this is now from Klein-Altendorf, a first result from imaging data collected this year, where we see again in the polarization properties a pattern that could reflect also the pattern of root distribution in the subsurface. and of course we need validation data to actually prove this, independent validation data, and so that's why we're all looking forward to the experiments we're now able to do within the FINROC context, where we will hopefully then not only prove the imaging capability of the method, but also then manage to address the issues of root function, root activity, and so on, as I was mentioning. And with this, I would like to summarize. I think I have 4, let's say, take-home messages, or 5. So first thing is crop roots. So I was only basically also tree roots do similar things, but here we're talking about crop roots. They exhibit spectral polarization properties, which are characteristic with regard to root structure and root state. And so sensitivity has been proven in several studies, I would say, but yeah, a model is not yet available actually to describe this. And this is my next point. So these mechanisms, so process-based understanding is very poor at this point, in particular including the spatial scale of the origin of the polarization that we observe. I mentioned that in the beginning that there might be contributions from the root cell scale and contributions more from the rhizosphere scale or whole root system scale. And obviously these models still need to be developed. Yeah, from lab experiments so far, we see that the method has capability, with limitations of course, for not only the structural, that is the imaging modality, but also the functional characterization of root systems. So, which of course is closely related to the point above, so to the understanding of the signatures themselves. But we can also apply this then in an imaging context. And yeah, the first results that we got from field experiments, it's only been a few so far, are promising. And yeah, we're looking forward then to the results that we hopefully will obtain within FINROC in this direction. And maybe from my point of view, the main challenges here are the development of this process-based Soll-Root electrical model from micro to macro scales. So I was saying, so basically if you want the cell scale to the root system scale. And then from a methodological point of view, the development, yeah, that should not be forgotten actually, the development of improved inversion schemes which adequately take the spatial, spectral, and temporal characteristics of root growth and function into account. Because I was saying it's an ill-posed problem. We have to actually, we have to come up with additional reasonable information to guide this imaging process, this inverse problem in the right direction. And that is computationally very demanding if we really would like to take 3D and frequency and time into account. Basically, we end up with a 5D inverse problem. And of course, then from a practical point of view, it's the implementation of the methodology for routing field sensing applications with many technical issues that I have no time to actually go through here, which in particular includes the endoscopic approaches, which we think could be practical from also from mobile platforms. And that's what we in particular also pursue within our sub-project in CP3. Yeah, and with this, I didn't hear anybody saying I should hurry up, so I hope it's okay. I have actually no watch here, right? I thank you for the attention, and I'm looking forward to questions.
Yeah,